Welcome to FDP Lecture on Pharmaceutical Biotechnology. This video will explore the evolution of biotechnology, focusing on how we are bridging the molecular gap from traditional heterologous expression to modern precision synthetic biology. The paradigm is shifting from exploiting wild-type biology to de novo genetic engineering pathways. Key chronological benchmarks illustrate this journey. It started in 1973 with recombinant DNA technology and the first active plasmid vector. By 1982, Humulin was approved, demonstrating industrial-scale cell factories. We are now reprogramming genetic information flow, intervening at multiple levels in the molecular biology sandbox. This includes optimizing replication, transcription, and translation, as well as introducing post-translational modifiers. Biotechnology is categorized by color based on its functional taxonomy. Red biotech focuses on healthcare, green biotech on agri-ecosystems, white biotech on industrial bioprocesses, and gray biotech on environmental remediation. This spectrum highlights the diverse applications of the field. The shift from empirical selection to synthetic design represents a leap in precision. Synthetic design uses in silico modeling and rational target editing for genetic manipulation, metabolic engineering, and optimization cycles, moving beyond stochastic methods. Classical recombinant DNA foundations rely on restriction endonucleases for cutting DNA, selection markers for tracking, and ligation kinetics for sealing genomic seams. These techniques are fundamental for constructing expression vectors. Recombinant human insulin production replaced immunological risk animal extraction. The process involves chemical strand synthesis, E. coli host fermentation, and downstream reassembly with chemical oxidative coordination to form the bioactive hormone. Recombinant monoclonal antibodies production utilizes hybridoma technology and mammalian bioreactor scaling. Techniques like somatic cell fusion construct humanization to achieve over 95% humanization, and CHO bioreactor runs are standard. CRISPR-Cas9 enables high-resolution molecular surgery by creating RNA-guided double-strand breaks. This requires a programmable spacer target in the sgRNA and adherence to structural constraints defined by the PAM sequence. Intracellular genomic engineering exploits two primary cell repair mechanisms: non-homologous end joining, or NHEJ, which is template-free and stochastic, and homology-directed repair, or HDR, which is template-driven and high-fidelity. A clinical application of CRISPR is the sickle cell therapy that targets BCL11A to reactivate fetal hemoglobin. The patient workflow involves cell harvest, ex vivo Casgevy electroporation, and autologous reinfusion following conditioning. Moving beyond double-strand breaks, we have precision systems like base editors, which perform single base pair conversions, and prime editors, which use PEG RNA complexes to rewrite, insert, or delete chromosomal arrays without triggering NHEJ. In tissue culture, plant cell totipotency allows reprogramming of somatic plant lineages. This regeneration cascade, controlled by hormonal stoichiometry, supports micropropagation for producing pathogen-free clonal crops. Marker-assisted selection, or MAS, accelerates breeding by tracking plant genotypes using linked genetic markers. It decouples trait assays from environmental variables and bypasses the GMO classification barrier. Plant genetic transformation systems include Agrobacterium tumefaciens, utilizing bacterial mimicry for integration, and biolistic particle bombardment, using high-pressure helium guns to shoot gene-coded microbeads into cells. In the realm of green biotech, Bt insecticidal transgenics provide pest protection. The process involves three steps: activation, proteolytic cleavage, and lysis. In lysis, the toxins form lytic pores that disrupt osmotic homeostasis. Golden rice addresses vitamin A deficiency by engineering endosperm carotenoid pathways. It utilizes a metabolic bypass loop involving genes from maize and Pantuia ananatus to restore the synthesis of beta-carotene, giving the rice its orange hue. Glyphosate resistance is engineered to overcome the shikimate blockade. Transgenic resolution involves sourcing the CP4 EPSPS gene, which has low binding affinities for glyphosate, providing complete crop safety. CRISPR-based gene drives overturn Mendelian limits by ensuring target propagation approaches 100%. Biosafety is addressed through control options, like split gene drives or programmable reverse mutation loops to mitigate ecological risks. White biotech process engineering includes enzyme immobilization to improve thermal limits and reuse, directed evolution arrays for selecting robust variants, and bioreactor aeration controls for real-time modeling of dissolved oxygen. Bioremediation frameworks use biostimulation and bioaugmentation strategies. Bioaugmentation inoculates target areas with specialized exogenous strains, such as Pseudomonas putida, to process complex chlorinated compounds into non-toxic end products. Systems metabolic engineering uses computational modeling to optimize product yields. Strategies include competing pathway knockdowns to prevent intermediate siphoning and lifting feedbacks to avoid pathway shutdowns from product accumulation. De novo synthetic PHA engineering focuses on marine biodegradable polymers. Polyhydroxyalkanoates, or PHAs, are microbial energy reserves. Pathway restructuring maximizes carbon flux to speed up polymerization rates, resulting in plastics that degrade in months. Bottom-up structural genome minimization, exemplified by JCVI Syn 3.0, creates a minimal synthetic lifeform. This chassis contains only 473 genes, providing a clean biochemical platform for predictable synthetic circuit operations. Advanced phytoremediation systems overcome limitations in traditional plants. Importing bacterial and mammalian genes, like the MIRA and 2 × 10⁻⁴ cypriot pounds genes, enhances heavy metal immobilization and volatile organic clearance in fast-growing trees. Frontier bioinformatics uses AI structural predictions to crack the protein folding problem. Deep learning networks, such as AlphaFold, predict 3D atomic structures from amino acid sequences, accelerating discovery from years to minutes. Scale-up bottlenecks arise because lab-optimized strains often yield less in industrial bioreactors due to nutrient gradients. Downstream recovery costs, up to 80% of total expenditure, demand maximizing cell titers and reducing purification losses for market viability. Biosecurity addresses the dual-use dilemma in research. Safeguards include digital sequence screening protocols to flag high-consequence agents, and built-in biocontainment engineering to prevent modified strains from surviving in wild environments. Bioethics distinguishes between somatic and germline editing. Somatic editing is non-inheritable and target tissue-directed, while germline editing is heritable, presenting complex ethical challenges regarding consent and altering the human gene pool. As biotechnology evolves, it drives a sustainable, circular bioeconomy. It offers tools, forward-coupling manufacturing from petroleum, optimizing ecological stewardship, and delivering transformative, curative precision healthcare platforms. This video has cited multiple external image sources across various domains. The links and sources are provided here for reference. Further image sources used in the video are listed on this slide.